concepts. What are the available dosage forms to have this particular target delivery achieved? So it is also called as a smart drug delivery. Why it is a smart means it is having advantages when compared to remaining doses with respect to efficacy and the safety. That is the reason it is very smart. So most of the regular conventional dosage forms, even though they are having efficacy, you bound to have some side effect along with the efficacy or the main effect of a formulation. Because it is a method of delivering the medication to a patient in a manner that increases the concentration of the medication in some parts of the body relative to the others. Because of that reason, we are having devoid of side effects and having less side effects and nil side effect, we can say that, because it is concentrating in only some parts of the body, because we are making smart enough to target them to some parts of the body. So most of the times we are using this delivery system targeting to a particular part, we are going to have it a nano level. So most of the times we are having a general tendency is a particle or any delivery system that is at the nano level, that is loaded with the drugs, and we are using them for the targeting to a specific parts of the body. So in this particular concept, these nanoparticles are the delivery systems acting as a carrier for the drug. They are just carriers. They carry the drug from the external to internal target site. So that carrier, it need to be smart enough to avoid any contact with any another body part except the body part what we wanted to intend to deliver. So thereby they are avoiding interaction with the healthy tissues. They will not be having any interaction with the other healthy tissues. Only it is having interaction with only diseased part. So what is the main goal of this targeted delivery? It is to prolong, another one is localize, and target, have a protected drug interaction with the diseased tissue, not with the normal tissue. Most of the times, the diseased portion may be either it is a carcinogen cancer cell or a tumor cancer or a tumor. It is required to be specifically targeted without getting affected with a normal tissue, because most of the times the normal tissue and the cancerous tumor tissues, they will be having a similarity, and it is very tough to distinguish between them. So we need to have a smart delivery system to characterize that difference and attacking the particular cancer cell without attacking the healthy cell. Similarly, even it is not the cancer, it may be having a particular defect at a particular organ level, cellular level, intracellular level. At the three levels, we are having a defects in our body. Because of that reason, we are getting a disease. So our smart drug delivery system in targeted drug delivery, either they attack the particular organ, because that organ only defected, remaining organ will not be getting affected. Similarly, it is in the organ also, we want to attack or target only particular cells in that organ. This is actually another level, that is the second level of our targeting. There is another level that is called as a third level. In the third level, what we are doing is we are targeting to intracellular parts like nucleus, your genes. So where we are delivering the drug to genes, that is actually considered as a gene therapy. It means our fifth and the first unit, targeting delivery of first unit, and fifth is the gene therapy, is the target delivery of a third order level is coming in the fifth unit. What we are using a carriers. What is that carrier? Most of the time they are having a nano size, and all these carriers are dealt and studied in the second, third, and fourth units. It means we are having aim in the mind in the first unit to at the goal at the fifth unit. We are reaching there as a path, second, third, fourth units. So the total together, we are going to achieve our target. This is actually what the journey in the molecular pharmaceutics. So first we will be dealing this particular delivery system, advantages and disadvantages, because you may be asked in 2 marks questions or 3 marks question in the session also. So what are their advantages and disadvantages, and the basic concepts are the different types of target drug delivery. The major advantages it includes, it is believed to improve the efficacy when we are targeting any site. Most of the times our aim is that our objective is also the same, to improve the efficacy. But at the same time, we should not have the side effect. How? The reduction in the frequency of the dosage form taken by the patient, that is one our objective. Having a more uniform effect of the drug by reducing the fluctuations in the circulating drug. This is actually more or less same of your novel drug delivery system. That is actually novel. Why? It is having a particular concentration which is required to that particular body part or a cell or a intracell organelle. So we need to have a uniform concentration which is reached for the desired period of time. By that we are reducing the frequency while reducing the side effects. How you are reducing the side effects? You are not allowing the dosage form or the carrier of the dosage form which is having a drug not getting attacked to the any another body parts or non-target organs. This is actually what our advantage of gaining this. So we are having a more efficacy, we are reducing the frequency, we are having a uniform effect without any fluctuations, so that the therapeutic window is protected without having a less than the minimum effective concentration and more than the maximum safe concentration. So we are keeping the drug concentration in the window, and we are reducing the side effect. By doing this, we are gaining the advantage of this target drug delivery. But the intrinsic disadvantage is it is having a high cost. Obviously, we are having this delivery system going to be high cost. But if you take pharmacoeconomics and study that, when you are taking the efficacy added with some money and side effects you are spending some money, all together if you are taking, and the total sum up, you are going to end with a beneficial effect only in the economy also. That is cost effectiveness also will be there. Even though the individual dosage form of a conventional and target drug delivery, they are having a differences in their original cost. If you see deep and study the pharmacoeconomics, they are highly beneficial systems. Even it is worth spending money to have that systems, because we are devoid of side effects and we are having more efficacy, and we are reducing the number of frequency of the dosage form, so that we are having a savings there, and we are not going any suboptimal concentration of the drug in the body where we are required to have that concentration appearing. Now we will be seeing there are two major kinds of the targeted delivery. It is you will be asked in the examination what are the different types of the target delivery systems, or you will be asked different targeting methods. There are so many classifications are present. So if nothing is being given clearly in the question, it is better to attempt all types of the classification, so that you are communicating that I know that the classification of targeted delivery in a totality. So if somebody is asking a specific active target delivery system and passive, say specifically by the name, you are going to deal them only. But unless otherwise it is not specified, it is better to make the concepts clear. So the major two methods are the active target drug delivery. Another one is a passive drug delivery systems. What is the difference between these two things? In the active drug delivery, most of the times it is a antibody related, that is actually receptor ligand interaction will be there. It is having a highest specificity and actively go to the particular target site, because it has got that particular specificity to that particular target organ or target cell or target cell organelle, because the receptor present in that particular organ or a cell or intracellular body which are having the specificity towards this particular antibody medication. So this is the reason it is specifically going actively there. So this is actually main advantage what we have made as an objective in the previous slide. Similarly, the passive drug delivery system, that has the enhanced permeability and retention effect, EPR effect. This is actually called as a passive drug delivery system. What is this enhanced permeability and retention effect? Most of the times when you are enhancing the permeability, what will happen? It will reach more concentration of the drug in a particular site, or that is organ or a cell or intracellular organ. It is having a highest permeability, so it is reaching there. So simply it is reaching there and it is having a reversal mechanism, it is coming back. Then what is its use? So for that purpose, what we are doing, we are having another mode of method is called as retention. So it is reaching there by permeability, and by some mode it is having a retention effect there for longer period of the time, so that we will be having a target nature. So target drug delivery purpose. It means in this particular approach, that is a method, the passive targeted drug delivery, every cell will be having a equal opportunity to receive this particular dose, and only the particular cell which is having a disease is having a more permeability and retention when compared to remaining. It means remaining healthy cells may also getting affected with this particular delivery system. It means passive drug delivery system are not that much safer when compared to active. It is appearing, but if you are having a highest EPR effect, then you are enough smart that even passive drug delivery system is taking your drug to only organ and a cell, or it is intracell organelle where it is having our target effectively with a greater retention, then it is equally as good as the top, that is active drug delivery system. Why you are doing this passive when active is that much specific is there? Because they are highly costly when compared to the bottom, where you are using a synthesis of your specific genome, and then you are making a transcription process, translation process, then you are getting an antibody, then you are making that antibody as a medication, or you are tagging that antibody with a drug, and that antibody taking the drug to the site, and you are making this journey possible. This is actually a time consuming process and a lengthy process and highly cost involvement and laborsome when compared to the bottom, which you are having a little bit safe, risky is there, but if you have got EPR effect is more, they are equally important and good. But this EPR effect achievement in the target delivery in the passive is the main target there. It will be there. Depending upon that only, we will see that which method I'm going to choose. It means if your EPR effect is good, then your passive delivery itself, it is going to apply for your treatment. Otherwise, you are going to have a active targeting. So in the passive targeting, the drug success is directly related to the circulation time with a nanoparticle that is between the 10 to 100 nanometers in the size. So most of the times, this is achieved by the adding a PEG to the surface of the nanoparticle. What is it is doing and making it to enhance the circulation time. So if you don't add this PEG to the nano delivery systems, what will happen to that? Because by adding this PEG component, that is pegylation, or adding a PEG to the nano delivery system or a nano carrier, what it is making is it is rendering it is more hydrophilic. So when it is hydrophilic, this is allowing the water molecule to bond to the oxygen molecules of the PEG by the hydrogen bonding. Because of that reason, it appears as a hydrophilic, and exterior moieties are already always containing getting hydrated. So because of this result, the bond is of the filmy of hydration is around the particle of a delivery system, that is a carrier, which makes the substances, what we are giving a carriers, antiphagocytic. So what is the phagocytosis? Phagocytosis is a phenomena where any foreign body is entering into our body, whether it is a nano or macro size. So the body attacks that particular particle and engulf it. Because of that engulfing mechanism, it will be getting removed from the circulation. It will not retain in the circulation for longer period of the time. So in order to avoid it, what you are doing? You are making it more hydrophilic. Because of that reason, it will be having an anti-phagocytic effect, because the cell is not engulfing that particular nanoparticle. It will have enough time for the permeation into the site where you want to target. Otherwise, before the journey itself, it will be caught by this police and taken to a jail, and it may be given a little punishment to that, and it will not reach to the target, because we have got a defense not to allow this particular drug to reach to the target site. So we are blocking that by doing this particular hydrophobic surface converted into a hydrophilic site, that is antiphagocytic effect which is having. So the particle obtain this property due to the hydrophobic interaction that are natural to the reticuloendothelial system. So the drug-loaded nanoparticles are able to stay in the circulation for longer period of the time. If you don't add this particular PEG to the surface, it will act as a hydrophobic. That a hydrophobic moiety is more prone to get having a phenomena of a phagocytosis. So it will be taken by the phagocytosis and it will be cleaved, and the drug will be getting degraded in the liver, and it will not be getting used for the circulation. So the target organ will not reach. In the target as active targeting, what will happen? The cell-specific ligands that will allow the nanoparticle to bind specifically to cell that has the complementary receptor. So this is actually something relevant to ball and socket mechanism, where you have got your paratope and epitopes. They are having some structural geometry that fixing specifically the particular receptor with the ligand. So the specificity takes the drug to the particular receptor which is present in the target organ, so that you will be having an active targeting taking place. For example, if you are taking a transferrin which was conjugated to the nanoparticle, so to target tumor cells that possess the transferrin receptor mediated endocytosis mechanism on their membrane, so the drug will be taken internally to the intracellular membrane by using this particular transferrin receptor mediated endocytosis. So it will be taken by specifically by this transferrin system. So it will be reaching to the tumors. So specifically attack it. This means that the targeting was found to increase the uptake as opposed to the non-conjugated nanoparticles. So if you are giving a nanoparticles without any conjugation of this transferrin, what will happen? You will be having access to every cell, but you are actively making a cancer cell to be getting engulfing this particular drug by attaching or tagging the drug to the transferrin, because the transferrin receptor, which will be having a mediated endocytosis, that will allow the drug to enter to the cell and reaching the target. So this is actually active target phenomena, where we are having an access to the particular drug to enter into a particular targeted site. So in the active targeting can also be achieved by utilizing the magnetoliposomes, that is physical manner. Previous one is a chemical manner, another one is a physical manner. Here you will be getting confused when you are categorizing or classifying the target drug delivery. So here you are entering into the physical phenomena of holding particular nanoparticle or carrier, that is like a liposome in a particular organ by using the electromagnetic principle. So thus by the grafting these liposomes with a desired drug to deliver the region of the body, magnetic position could aid with this particular process. So you will be having an external magnet with that influence the internal magnetic attracting species like a iron species which you are adding there, that will take the drug to retain it in the field where it is more having magnetic effect there. So it will get retained. So externally you are guiding something to load in a particular actively reach the target organ by the external magnetic field. This will guide the nanoparticle to be getting concentrated in a particular specific site actively. Even though it has got access to all, but it will be actively present there because of this external your magnetic field. So positioning system. So this is actually helping. This is another physical mode. So then you can have another mode of making your classification also is called as the inverse targeting. What is this inverse targeting? How you are making the same name what you are using for the passive targeting? Don't get confused. Nothing but the inverse targeting is against to the active targeting. It is same as that of your passive targeting. What it is doing? It can be achieved by the surpassing the reticular endothelial system by the pre-injection of the large amount of the blank colloidal carriers and the macromolecules like dextran sulfate. All the endothelial system is busy. When it is busy with the blank molecules, then you are making them not to discover your drug-loaded nanoparticles, so that you will be having access to reach the target organ. Otherwise, they will be getting engulfed by the phagocytic manner as a regular your passive targeting only. But here you are avoiding because you are making the reticular endothelial system busy by giving a blank colloidal carriers which are having a similar capacity to attract your reticuloendothelial system. So it is based on the avoiding the passive uptake of the colloidal carriers by the reticuloendothelial system. In other words, it is a reversion of the biodistribution trend of the carrier, and the alternative strategy is including a modification of the size, because each organ has got a specificity in the size of capillaries. So we are using a specific size nanoparticle to reach that particular organ. And surface charges, most of the times it is depending upon the disease state, it will have an alteration. Accordingly, we are having and the composition and the surface rigidity, because of the differences we are having, like what you have seen previously. The differences in the surface rigidity, because of that permeability will be altered with the tissue, whatever which is present in the target organ. Most of the times in some cases you find that in the diseased state, the surface rigidity of the target organ will be less, it will be more flexible to make the drug entry more ease. In that case also, say for example, in the meningitis and blood-brain barrier will get disrupted easily to enter the drug concentration when compared to normal blood-brain barrier. So these effects will be present in those cases also. You will be bypassing this reticuloendothelial system in early stages and making the drug available large concentration at the target site. And the hydrophilicity characteristics of the carrier, this we have already seen that we have added PEG or pegylation to your nanoparticle surface for the desirable reaching and biofit of that particular moiety by altering the hydrophilic nature of the surface. So most of the times the Ansrami has already reported that the phospholipid microsphere emulsified with poloxamer 338. This is a hydrophilic like a PEG, which is present, what you have seen in the passive targeting pegylation. Here he has used the first person to use in 1985 and found that the lowest reticuloendothelial uptake is present, that is used for the inverse targeting. So you are making the more concentration in the target site, because it is a hydrophilic non-ionic surfactant. He has used it. Later onwards, PEG been chosen and more studies have been found, and this is effectiveness of bypassing this, and it is being used. So it is almost same as that of your passive targeting only, and you are making this type also useful, but it is not that much worth as a active targeting. Now we are coming to the another class of classification is a physical targeting, another one is a chemical targeting. So in the strategy, using the size, shape, charge, stiffness among the other parameters to influence the tissue accumulation and adhesion and the self-actase is coming under the physical targeting. Like your magnetic microspheres or the nanoparticles or liposomes, they are coming under this, and the sizes of different nanoparticles linking to your capillary size of your organ, that is another one, and having a flexibility and making a rigidness as the index to enter into the target organ. This is another parameter. All the physical parameters which will be affect your targeting, that is actually called as a physical targeting. Another one is a chemical targeting. It is based on the molecular recognition, like your receptor. It is coming under the most active targeting, where you have seen the specific receptor interaction units to. Most of the times what we do, your drug is having not a specificity, we are tagging by decorating the surface of the particles or the molecular units that will have a responsive to the diseased environment, that will be having a specific simulative to attract it. Then we are taking the drug also there by attaching or covalently bonding to the particular moiety. Now we are going to see the different levels of targeting in classification of targeting drug delivery systems, and you will be having the different concepts which are linked to these types only. We have got a first level, second level, and third level. So 3 levels are present. In the first level of targeting, our target is at the target organ level. So it is whole body is containing a different organs, and we are taking first level of targeting to reach our drug restricted to the particular organ and not getting affected with the remaining healthy organs. But because we have got a problem, a cancer in a particular organ only, we want to attack it. We have got a problem only in a blood-brain barrier, that is a brain only, we want to attack it, not other parts. So our target and the first level is the organ level, we have got a targeting. Similarly, we have got a specific organ which is having a problem is a liver, and liver cancer is present. We want to target only to the liver. Coming to the second level of concept of targeting is targeting to the specific cell which is present in that organ. Like you see Kupffer cells which are present in the liver. So they are also a reticular endothelial protective cells. So they will be getting attacked just like your reticular endothelial system protecting layer what cells which are floating in the blood. It is also having a cells in the liver. They engulf it. So in this particular case, you will be having a specific cells only getting affected when compared to remaining organ cells which are present. So it is in the second level, it is attacking a specific cell in the organ. In the third level of targeting, we are entering into intracellular level. In all cell, the whole cell will not be getting affected, but the drug along with the carrier is entering into the cellular cytoplasm and reaching to the target. It may be a gene. And we are going to use it for the gene therapy. That is actually third level. It means the old concepts what we are learning in the first unit, we are reaching to the third level in the fifth unit by using second and third, fourth units to take the carrier reaching there. How we are making your drug target to that particular carrier reaching so that it is having a targeting effectiveness. Otherwise, we will not be able to get this reduction in the side effects. So this is actually what the levels of the targeting. To achieve this, we are having a ligand and antibodies. For the active specific targeting, we are having a ligands and antibodies. They have got a specificity. Because of that reason, our targeting index will be very high. So our intensity of the drug action will be very high in the target organ. To achieve the high specificity, nanoparticle can be conjugated with the targeting moieties on the surface that bind specifically to the receptor which is present in the organ or cell or intracellular organ. So this is coming under the chemical targeting. Don't get confused. The classifications are getting interacted. Till now, what we have seen is a physical target. This is passive targeting and active targeting, physical targeting and chemical targeting. So now we are entering into the specific targeting. We are having ligands and antibodies. We are seeing there in the ligands and antibodies are coming under the chemical targeting of your active targeting. So this one you need to remember. Examples of the chemical targeting is via antibodies are the ligands that improves the delivery. Most of the times we are having a carriers like a PLGA. So we will be preferred polymeric system because it is recommended as a safe and it is used for the internal purpose for the because of the potential of biodegradation and approved by the FDA. Polyglycolic acid. So it is actually L-glycolic acid. It is not the D. So this polymer which has got a biodegradable in nature and simulated to our body mechanisms and which can be proved as a safe and it got validated also and it is being used and so as a biodegradable polymer we are using them and the nanoparticle can be absorbed or conjugated with the antibodies on its surface by having this interaction of a covalent bonding or a bonding with the antibodies. So they will be having different antibodies like a biotin. This will be used for the streptavidin. So you will be having a thiolation to have a bonding, making antibody attached to this particular surface. And the nanoparticle may be getting interacted with the drug. So this tagging taking the nanoparticle to the desired site. Within the nanoparticle, you have got a drug at the target site. They will be getting attached there in the ligand and making the slow release of the drug at the controlled manner there in the target site. So it is taking a longer hour of presence so that you will be having more concentration at the target level, target site only without having any effect on the non-targeting site. So the cross-linking agents such as bis-sulfosuccinylate, imidyl subarate, these are all used thiolation mechanism or cross-linking mechanism. These require to be non-toxic, non-irritant, should not affect your drug and your carrier and your body parts. That is the reason you need to choose this either any mechanism. At the same time, it should be reversal or it should be biodegradable, getting attacked with the body. At the same time, it should be long enough to get withstand in the vicinity of the target. Accordingly, we need to choose the mechanisms. Most of the time, stimuli responsive systems, either it is a physical or a chemical stimuli, which we are going to use for the targeting, in addition to your regular ligand based or receptor based targeting. So most of the time, stimuli responsive strategies in the targeting and the concept of drug targeting, if you want to know it, any one of these 5 can be chosen with a preferably depending upon what disease we are getting attacking. So what we are choosing the system, the first one is the pH. Acidic compartments found at the pathological tissues, especially in the tumor sites, are within the cells in the endosomes in particular diseased conditions. When compared to regular, your pH of your body is 7.4. So this is maintained, but in the diseased conditions, in that particular part, you may be having an acidic compartment. That specificity of the particular carrier to that particular pH, lower pH, is actually a strategy to act as a stimuli to reach there. This is actually one type. Another one is a thermal stimuli. How to target in the thermal stimuli? We are finding a heat applied externally if the tumor is near to your skin. It can be possible, or the physiological changes which are occurring, maybe a cell synthesis at a particular tumor is more, and a localized thermal changes may be occurring there, that can be using as a guide to reach the target. So this is actually one mechanism, thermal mechanism. Either it is internally induced or externally induced. There are 2 types now. Internally induced means that tumor or a target organ itself, it is having a little bit changes in more temperature when compared to regular tissue because of the more cell growth which is having that, because of those changes which may be having effect. Or external stimulus we are applying, but this cannot be used for the interior tumors, but we can apply for the external stimulus for the exterior tumors, which can be accessible. Light, this is actually externally applied light most of the times, which we will also be getting used to have a photolytic changes internal the target organs and making the target reached by the drug. Another one is the redox reactions. So most of the times region where the reactive, oxidative and reductive species are abundant, most of the times in the inflamed tissues are the tumor tissues. Because of these changes in the redox conditions, we are going to use it for the targeting. Another one is enzyme. Regions of the high protease expression in the cancer tissue is present when compared to regular tissues. So this protease expression, it is acting as a stimuli to have a drug targeting. So you have got a specific ligand or the receptors based, or you have got these 5 stimuli based systems for the drug targeting. This concept will make you to select which system is good for me to have the targeting achieved with least cost and fast enough and effective manner. We need to choose it. This is actually what our formulation scientist, we need to select which targeting delivery I need to select. Treating the brain diseases. Most of the times, this is actually what the hurdle which are taking place in the psychiatric disorders and psychotic diseases and tumors which are present in the brain, or a specific cellular or hormonal imbalance which are taking place at the organ, that is brain. It means we are targeting to the brain, we are taking this one as an organ, then it is coming under the first line of targeting. Say for example, if you are taking only a particular part in this particular brain, like you are taking a gland in the brain, a pineal gland or a growth hormone changes you want to make, then it is going to a particular part in that organ, then we are going to a cellular portion second level. In that case also you are making a changes at the gene level and a gene modification you want to make it there, then we are going to have a third order. But in all the cases, your reaching to the brain is a major problem because your blood-brain barrier, it will have a highly barrier effect. That is the reason that barrier effect is more predominant. Even first order targeting itself, first level targeting itself, very tough. So once you are having a first level targeting is there, when you go to the internal to the brain, then you can think of second level and third level at the intracellular level. So this is a major hurdle which are present in this particular targeting to the brain. So why this challenge is appearing in the brain, you need to understand so that you can attack in a detailed manner later. So the structure of the blood-brain barrier capillaries of the brains are lined up with a layer of special endothelial cells that lack the pores. They don't have any pores there and are sealed with the tight junctions. And this is one bigger barrier, so that is making a trouble to reach the brain. Why it is been made? Why God has created like this? You may be having an answer that because it is the nervous system for the functioning of entire body. It is controlling autonomic and parasympathetic systems which are present. Sympathetic systems, the entire thing is controlling the body. We cannot allow any external influence to be having a changes in our function or the growth of our survival. This is actively making a survival process at the stake. If you are allowing the blood-brain barrier is not there, then existence itself will become a problem. That is the reason this has been made as a defensive mechanism by nature itself to have that particular tight junctions and not to allow any external matter to enter into the brain. These tight junctions are called as zona occludens. So the zona occludens, it will actually making it tougher to not to have any pores and reaching the brain, it making it tough. So the tight junction produced by the interaction of several transmembrane proteins, they are called as a occludin and claudin. So this is making a tight junction that project into and seal the paracellular pathway. So we don't have any pore transport into the brain. So you will be having a pore transport completely nil, unless otherwise you are not using any carrier mediated transport, that is active targeting. It is very tough to reach the brain. So the interaction of these junctional proteins is very complex and effectively blocks the aqueous route of the free diffusion or the polar solute from the blood along these potential paracellular pathways. So that denies these solutes free access to the brain and making intestinal fluid. So brain interstitial fluid, that is the extracellular fluid, that is it is very tough to making to reach there. Further purpose, what we are doing in the targeting is two approaches we are going to use for the targeting to the brain is invasive and non-invasive methods. So invasive method means we are reaching the target physically by injecting a needle and invading it and making a drug delivery system reaching to the site. This is actually what the invasive system is. The second one is the non-invasive method. We are not going to touch the physical route and making without having a disrupting by inserting the needle, we are going to use it. So some routes of the direct administration of the brain are non-invasive, such as the transnasal route. So the it is been also been studied, but it is under the stake and still problems are still present. How effective the transnasal route is required to be clearly studied further. Whereas others involve the entry into the CNS by the devices like needles. So that is such as in the case of the intrathecal and the intracerebroventricular is considered along with the sustained and controlled release of the delivery. So to reach there and injecting there in the particular site is another approach. And direct injection into the brain. Most of the times, even you are taking your drug to the cerebrospinal fluid, but we don't know how effective that particular drug to reach the brain is still under question, because even the cerebrospinal fluid is required to have a barrier effect to the some of the components which are present. What you are giving injecting a drug into that one to reach the brain. That is the reason a third approach is called as a direct injection to the brain. So you are not given into the cerebrospinal fluid. So first thing is non-invasively entering the brain. The second one is reaching to the cerebrospinal fluid and entering into the brain. The third one in about 2 hours it is becoming a still barrier effect. So we are giving a direct injection to the brain. These three are the strategies. which are including invasive and non-invasive methods. So the last one is actually direct injection into the brain is a invasive method. So that is actually which we are required to have so many studies, and the clinical interventions are required to be studied. So how effective it is there, because the internal brain reaching by the direct injection is not suggested, because we need to cut the skull, we need to cross so many brain tissues to reach the internal midbrain structures where the disease portion is there. So it is actually a lot of research required to be done at the clinical level. So to do this particular route of direct injection by the invasive method. So this is they are required to be greatest development required to be anticipated to occur the area of the drug targeting by the systemic administration by the direct injection. So how we are going to do them? So these invasive method and non-invasive method, still they are having a problem and advantages and disadvantages. How can I go approach for the targeting to the brain? So there are three methods are present. One is manipulating the drug lipophilic analogs and the prodrug carriers. So instead of giving a drug, we are converting a drug to the prodrug so that it is having a better affinity to the particular receptor, or which is having a better permeability index, so that we will be having a targeting potential and retention potential. So this is one mechanism. The second mechanism is the disruption of the blood-brain barrier. So that is the osmotic biochemical method. So we are going to have a reversible disruption. Irreversible disruption is actually changing the brain structure. It is not suggested. So disruption for a particular period of the time. After that, again coming back to the normalcy is required. And the third one is the alternate routes of the CNS drug delivery systems by the intraventricular and intrathecal. So where we are going to explore. But it is required to be simultaneously having some active targeting systems like your ligand or the receptor based mechanism should be accompanied in the third method. The first one is the lipophilic analogs, CNS penetration by the low molecular weight lipophilicity requirement. For that purpose, a liposome has been taken as a carrier for the targeting. So the drug targeting carrier is a liposome, where we are entrapping a doxorubicin within the hydrophilic environment of a within the liposome, or it will be having amphotericin B. They are highly toxic moieties and having a greater side effects. And we are going to entrap them in the hydrophilic portion of liposome. The external portion is having a lipoidal in nature, so they are having highest permeability index so that they can cross. This is actually a lipophilic analogs. So there is another drug which we are making is we are going to have a prodrug approach. We are having some drugs which are converting into original active moieties after having a reaction with the enzymes which are present within the brain system. This concept is also good. So the prodrug improves the physicochemical properties, that is like esterification or amidification or hydroxy or amino or carboxylic containing groups, enhance the lipid solubility if you are leaving them as a ionic species. So the penetration will be very high. We are going to convert that electrolyte formation or the ionic species to non-ionic species by having this esterification and amidation. So that will cause the enhances the lipid solubility of the drug and re-entry into the brain. Within the brain, they will be having hydrolysis by the enzymes, and the active drug is used. The example is the levodopa. So we are having for the administration of the dopamine, we are having a codeine for the administration of the morphine. So this is a specific example which you can alter the drug to have the crossing of a blood-brain medium. The carrier mediated drug delivery systems where the drug acts as a pseudo nutrient that undergoes the carrier mediated transport. So we are similarly making a carrier, the drug moiety, which is same as mimicking the transport, or we are making a antibody. So that antibody is having a specificity to get mediated. So we are tagging that to a liposome or a targeting carrier like a nanoparticle. Along with we are having a specific drug. So here we are having different transport systems for the transfer of nutrients to the brain, like a transport system for the nutrients like amino acids, transport system for the monocarboxylic acid, we have got a transport system, nucleoside transport system. These are all getting used as a carrier to reach the brain. So these carriers which we are going specifically used by having a interaction with these receptor by the material, that material is having a specificity to that particular receptor. Along with that, you are having one drug which is entrapped within that particular carrier. So we are reaching the brain. This is actually what the carrier mediated in the brain drug delivery. So these are all the basic concepts which you are learning now. And further depth we are going to see in a little bit later what I mentioned. The fourth one is a disruption of the blood-brain barrier. Here we are using the osmotic disruption by the intracarotenoid injection of an inert hypertonic solution of a mannitol, that will mannitol or arabinose. So that results in the endothelial cell shrinkage because of the hypertonic solution. You know that hypotonic and hypertonic. Hypotonic, it will make the bulging effect by making a expanding your cell membrane, and it will cause the cleavage. That cleavage is not required and it is not suggested at the blood-brain barrier level, and it is not that much easy also. And that is the reason we are using the hypertonic solutions to cause the shrinkage and opening of the barrier because of that shrinkage and opening the tight junction for the few minutes and for the delivery of the drug to the brain. So this is actually a strategy. Now, the blood-brain barrier, we are reaching to the particular site, we have crossed it. So we need to have a crossing of a specific organ level within that, that is a specific level 2 targeting. So that potential of the targeting chitogen nanoparticle to the brain using the mouse monoclonal antibody for the carrier mediated transport is already been used against the rat transferrin receptor system to enter into the brain. It was successful. Then it was thought that as it is a receptor mediated delivery for the brain targeting is what considered. So the transferrin system receptor has been reported to be high abundance in the brain microvascular endothelium. So this will be used very much in the brain targeting for the cancer in the brain. So we are taking a transfer of this transferrin mediated carrier. So that is reaching to the blood-brain barrier, cross it, and reaching to the internal brain structures. The goal was inhibition of the caspase 3 like protease activity, which is encapsulated by the N-benzoxycarbonyl DEVD fluoromethyl ketone peptide. Because of the therapeutic significance in the treatment of the stroke and related neurological disorders, which are not present to the exterior to the brain, it is entering into the brain stem stage. So the carrier mediated transport only is required to reach these receptors. So that is the reason which will be having a transferrin receptor mediated drug delivery. So the strategy involved the use of the biotin labeled PEZ to coat the chitogen nanoparticles and streptavidin labeled antibodies for the conjugation. So this conjugated nanoparticle can penetrate into the brain. So this is actually what the strategy is used in the initial stages, where you have got a success criteria more in the case of the rats, and the same thing which is proven to be working in the human being also in nearby future, but still more studies are required to be done to make it happen, but still it is under the question mark. Now targeting to the cancer cell, this is our syllabus portion, which is having the second portion of a targeting is a targeting to the cancer. There are different types are present. What we have already seen, 5 response systems are most of the times which is allowing to targeting to the cancer. The first one is a pH responsive. So the most of the body has a consistent neutral pH. Most of the times it is having a 7.4 near to the neutral pH. However, some areas of the bodies are naturally having a acidic, then it means it is having a lesser than 7. But the body is having 7.4. Thus, the nanoparticle can take an advantage of this ability to reaching the drug when it encounters the specific pH, that the pH responsive systems are going to help. The second approach for the treatment of the cancer specifically is based on the redox potential. One of the side effect of the tumor is a hypoxia, because they are having the highest cell growth and consumption of the energy is very high, and oxygen consumption is more because of the profuse cell growth. So it is resulting in the hypoxia, lowering in the oxygen concentration, oxygen potential, which alters the redox potential in the vicinity of the tumor. So by modifying the redox potential, that triggers the payload release of the vesicles that can be selective to release the drug in the specific tumor only, not in the normal site. Because in the normal site, you have got a hypoxic conditions, and the potential redox potential is different than your cancer cell. So the drug will not be released in those areas. Only in the hypoxic site only it will be releasing, and it will be having specifically in the tumor. This is actually what the strategy is to target to the cancer. Most of the times there are so many series of studies have been done for the having stepwise involvement in the reaching a target site of a tumor. So one is epidermal growth factor receptor. This is actually used for the active targeting for the tumor. When compared to your stimuli based, it is that is actually passive technique. In this active technique, a receptor is highly expressed in the breast cancer, EFGR. So it has been used with PLG nanoparticles and containing the rapamycin. So that is actually the drug that is surface conjugated with the EPFGR antibody. So this is actually, this antibody is taking to the particular cancer. So that is tagged with your drug. So your concentration in the breast cancer is going to be very high, and effectively it is getting treated. Antibody conjugated nanoparticle can also be used for the targeting delivery of a nucleic acid delivery. Next PowerPoint, we are going to see that there we are going to have, instead of your using your amino acid sequences, we are using a nucleic acid sequences to reach the specific site and having a specificity to have a specific responses in the within the cell. So for that purpose, we are going to use a smart interfering RNA. It is actually a small. Why it is called as a small RNA means it is having a nucleotide in the range of 20 nucleosides, 2021. It is a this small interference RNA. It acts as a therapeutic. Has been shown a lot of promise. So, but the main obstacle is preventing them to degrading upon the delivery, because in the travel itself they will be getting cleaved by the hydrolysis. So that even can be done by the CALAA01, a nanoparticle formulation, which has been already been studied with a code to deliver the small interferon RNA in the tumor cells. Is the first nanoparticle of this particular type entering the human clinical trials to reach the intracellular cytoplasm and reaching to the nuclear site for the effective targeting. That is actually a third targeting. So now we have come out of some of the things, and we are going to enter into the next level for the further advancements. There we need to learn some more questions and concepts, because you will be asked in the cancer cell or targeting to the brain. So you will be asked what is apoptosis, and apoptosis based drug delivery systems are the treatment. For that purpose, you need to know some of the concepts. Then only you can answer those questions. So this is actually something which you are having a terminology, and based on this terminology, we are using these active and passive strategies, or you are going to use mechanisms or approaches like a physical or chemical methods to treat our diseases by the targeting. So what is apoptosis? It is a form of programmed cell death that occurs in the multicellular organisms. It is intrinsically a nature, it is providing it. So the biochemical events lead to the characteristic cell changes, and it is causing the death, and it is taking a rebirth, and it is a normal phenomenon. So this is actually a programmed cell death is called as apoptosis. So the changes include the blebbing, shell shrinkage, nuclear fragmentation, chromatin condensation, and chromosomal DNA fragmentation, and global mRNA decay, and ultimately the cell is getting dead, and it will be taken by the reticuloendothelial system, maybe entering into the liver and spleen, and it will be going out of our body. After that, a new cell will be getting generated in the place of original dead cell, and body is getting rejuvenated. This apoptosis process is actually a normal phenomena which is required for the rejuvenation and normalcy. Most of the times, what is going on in the apoptosis is mediated by the proteolytic enzyme called the capsages, which trigger the cell death by the cleaving a specific proteins in the cytoplasm and nucleus. So the capsages exist in all the cells, and inactive precursors are the procapsages, which are usually activated by the cleaving by the another capsages, producing the proteolytic capsase cascade. So this is actually, it means that the cell dying mechanism is already present in our body as a procapsase, and it is going to work after a particular trigger. That means it is just like we are giving a tagging of expiry date to our medicine. So after the expiry date of the cell is there, this procapsase will be cleaved to converted into a capsase, and this is going to cause the cleavage and producing the proteolytic cascade. The cascade means series of reactions to cause the cell death. That is a cell shrinkage and cell death. This particular phenomena is actually which is absent in the case of cancer cell, or it is going to trigger having a more cellular growth and making a cancer cell growing. So we need to consider these phenomena and approaches for the treatment in the cancer subsequently. We are going to have a targeting for the vaccines. So this is actually what